lab procedure, how do we actually deal with these kind of samples. Everything will be presented today. So yeah, so that will be there, and I really hope that today people have more questions. So please, guys, again, I'm saying if you don't understand anything, please stop me and ask immediately. And I am also assuming that you guys are taking notes, okay? Because I don't think that everybody are shared with this slides right away. So make sure that you take your notes as we go through the sessions, because even if you save the presentations with you, remember that everything is not there in the presentation what I'm going to talk about. So whenever I'm making some important points, I am hoping that you guys are taking the important points or noting it down somewhere. So please do take your notes. Okay. So let's start with introduction part. Akhilesh, please next slide. Okay, so let's start with the introduction. So, what is lysosomal storage disorders? Okay, so first of all, let's see what is the purpose of the test. So, here we'll be ruling out lysosomal storage disorders, and these disorders, as I just said, that these are uh, because of some kind of deficiencies of enzymes that are functional inside lysosomes. I will also tell you what is lysosomes. So basically, whenever there is some malfunction of some enzyme which is actually working inside the lysosome, we will see cases of lysosomal storage disorders. See, as the name suggests, that means disorder is related to something happening inside the lysosomes, and these disorders are storage disorders because there is something that is getting stored a lot than normal, you can say, and that is why we call it as lysosomal storage disorders. Okay, so what we do in this is we just monitor the enzyme activity of whatever enzymes that are involved in these disorders, and there are a number of disorders that you will be finding in this category. I mean, there are almost 50 disorders, okay, but actually there are more than 50. But also remember, these are kind of very rare disorders, and out of those 50 disorders we rule out 30 most important and most commonly found disorders in the populations, you can say. Okay, so these 30 disorders majorly covers the most important or the most crucial kind of enzyme dysfunctions that are observed, and we almost cover all of them which are like important. Okay, and as said yesterday also, please remember this name. The instrument that we use is a multimode reader called Varioscan Lux, and this is a production of Thermo. Okay, here we'll be again using the principle of light. Yesterday, if people remember, I told what is principle of light. Principle of light is nothing but there will be a involvement of different wavelengths that are showing some signals at different. Again, these are the light is like it can be a visible spectrum, invisible spectrum, and these light molecules will be behaving in a certain way at a certain wavelength, and that is what will generate a signal showing a presence of disorder in patients. Okay, next slide. Okay, so as I said, I just want to show you what is lysosomes. So basically, lysosomes are kind of a cell structure. Like basically, you guys know that human body is made up of a lot of cells. Okay, and cells, another thing, it will have a lot of units inside it, right? So lysosomes are one of the cellular units that are like a vesicle, or they are in a shape of a sphere, first of all, and they are also, you can also call them as a cellular sacs or vesicles. And what happens in lysosomes is they're also called as the suicidal bags. I will tell you why. So what happens in lysosomes is that they are filled with hydrolytic enzymes. See, whenever I say lytic enzymes means enzymes that can lysis itself means cutting down something. Okay, lysis defines as something is getting cut down. So what these enzymes are doing now is they are cutting down the you can say certain things, or they are reducing larger molecules into smaller units of them. So what
Breaking
an
down.
sorry.
Breaking down, no?
Yes, breaking down or cutting down, you can say a large kind of a molecule into its smaller units. For example, if there are some fat cells or some fat molecules or some lipid molecules, so these are very large molecules, okay, and the body will not directly utilize it. Whenever we eat something, we do not take direct forms of glucose, right? We take different types of proteins, carbs, and lipids and fats, but the body is not functional to directly use such molecules. So what these units will do is, that is lysosomes will do is, they'll just have the ability to break down these molecules and make convert them into simpler sugar forms so that the body can use it. Okay, they turn them into the simpler versions of whatever you are actually consuming or whatever goes inside the body. Okay, so next slide, please. Okay, so that is what was the basic thing happening in the lysosomes. Now we'll see that what happens if there is defects in one of the enzymes. So basically, when I say that the lysosomes break down the molecules, it is not the lysosomes itself. There will be some enzymes present inside those lysosomes that are actually carrying out the activity. As I said, there are hydrolytic enzymes available which will be um, breaking down these biomolecules. Okay, so now let's see what happens if there is any defect in lysosomal enzymes at all. So what happens is, these lysosomes will contain enzymes responsible for the breakdown of various biomolecules, and defects in the synthesis leads to lysosomal storage disorders. If one of these enzymes are defective because of mutations in coding gene, that means, see, whenever I say that these disorders are caused because of some enzyme defects, do remember that this is not some externally present disorder. That means there is no bacterial infection or anything that will lead to these disorders. These are all genetically present disorders. So when I say genetically present disorder, that means there is some gene that will be always involved, and whenever there is a problem in these coding genes, I mean they are not correctly functioning, or there is some kind of mutation in these kind of genes, that is where the enzyme can be defective, and hence it won't be functioning properly, so that you can actually get a normal function, right? So as I said that these enzymes will be breaking down the biomolecules. What happens is, due to the defect in the enzyme present, these biomolecules start to accumulate within the cell. Okay, instead of becoming the smaller units of its own, they stay as they are. For example, there's a kind of fat molecule that you are consuming or that is going inside the body, but it is not going to break down into its simpler units. Then what will happen is, they'll just keep on accumulating inside the cell. Okay, now I have just for you guys to make it simpler, I have attached one picture here. You can see that a healthy cell has a certain procedure. In the first image, actually, you can see that whenever there is a molecule entering inside the lysosome, so they are breaking down and utilizing whatever is required by the body. The unnecessary parts are eliminated and some parts are recycled. That means they will be stored and used again if required. But if there is a presence of disorder, as you can see in the second image, the these biomolecules are getting stored. For example, as I was saying, if there is a lipid molecule and it is not going to get broken down, then there will be such kind of accumulation. If you can see in the second picture, you can see that there is a lot of fat, you can say, present inside the cell. Now, why this is important is, it's, for example, I'll just give an example of a balloon. When you blow a balloon can be only blown until a certain point of time. That means if you fill too much air in it, it will eventually burst. So the same thing will happen with the cells. If these molecules are not getting recycled, they will start accumulating inside the lysosomes. Too much quantities will start accumulating inside the cells, because once the lysosomes are defected and they're not able to break down, these molecules will start accumulating in the cells, and eventually there will be a time when they'll start destruction of the cells itself. Hence, it can be a very fatal condition also, and hence these disorders are to be taken very seriously. Okay, most of these disorders are rare and autosomally recessive. As I was just saying, that these disorders are not some kind of bacterial or viral infections, that they are coming from some outside source. They will be inherited genetically via parents or some family lining. You can say that there was some genetic mutations, and hence these disorders are inherited in the younger generation or the future generations, you can say. When I say genetically inherited disorders, for people who don't know what that means, is these disorders come from parents. The parents are normal because they are just partially affected, and when someone is partially affected, they do not necessarily present any symptoms of the disorder. But if those defective parts, half defective part of a mother and half defective part of a father, comes in a child, then obviously that half and half makes, they are completely affected, we will be calling them as affected individuals. Okay, so that is what you can say is genetically inherited disorders. And as I have told before, that these disorders are very rare and they are autosomally recessive. Actually, these are the genetic part of it. I do not want to speak too much about it. Later on, as the sessions go, you will understand all the genetic sides of it also. Okay, with the next upcoming sessions, you can actually understand everything, but please do note whatever important terms are there, as I was telling yesterday as well, that you can read about it whenever you have time. The terms that are new for you, please do read about it. Okay, and next is that we classify these disorders depending on what kind of accumulation is happening inside the body, and what kind of enzymes are showing some defective activity or some deficiencies, which is leading to the disorder. Okay, next.
Uh, just 1 second, Divya.
Yes, tell me, Devendra.
Yeah, just please go to last slide.
Akhilesh, please go to last slide. Huh.
Yeah, in the first sentence, lysosomal contains enzymes responsible for the breakdown of various biomolecules. The effects in this synthesis leads to LSD. Synthesis
Hmm.
means, is this just the process of breakdown or it is something
Yes,
else?
yes. No, synthesis here means the process of breakdown. That is what it means here.
Okay. Thank you.
Oh, next slide, please. Okay, so actually, I have purposefully added some picture for you guys to just understand, like, what exactly I'm trying to say is happening inside the cells. Okay, so you guys, if not by words, you can at least understand by the pictorial level that what exactly is happening inside the cell, leading to its destruction. Okay, so as I just told you, that depending on the type of accumulation that is happening inside the cells, we will be classifying the disorders depending on that. Okay, I am very well aware that these names will be very long for you guys to understand. I do not expect you guys to understand immediately, but please do remember try to at least remember that what are these molecules called. Okay, here I have distributed them into 7 categories. Okay, and these categories are also like here when I, for example, the first category is sphingolipidosis. So in sphingolipidosis, there are molecules called sphingolipids that are getting accumulated inside the cells. Similarly, the second category is mucopolysaccharidosis. I know again these are very long terms. Please
Ma'am,
break
please
them down.
repeat twice, ma'am, at least we can understand the pronunciation of the word. Repeat twice,
Okay.
ma'am, please.
So that is what I'm saying. If you don't understand exact pronunciation, you just have to break down the words. Okay. For example, the first category is sphingolipidosis. So just break it as spingo, then lipid, and then osis. Sphingolipid is a type of lipid that is getting stored, and osis is basically a term used to show presence of disorder. Okay, so disorder present because of sphingolipids.